All right, let's talk about a couple of ways how cell can regulate the expression of enzymes. One of them directly connects to what we were talking about just a few minutes ago. So this is the concept of riboswitches, which some of you may never heard before, okay? And yeah, at the very, very detailed molecular level, it can be very complicated, but conceptually, it is really a simple idea. So what is the riboswitch? And I'm not even going to draw the entire structure. Remember when we were talking about RNA, we mentioned that RNA forms inside of molecule, intramolecular base pairs, forming that mushroom-shaped hairpin loop structure, like this. Okay? So these hairpin loops, one of their function to serve as a riboswitch. Okay? So schematically, this is the riboswitch in what we call an on position. Okay? There can be two outcomes. Ribo refers to RNA. That's important. Riboswitches are RNA elements. So if riboswitch in the RNA molecule is on, RNA participates in two gene expression steps. RNA is the product of transcription, and it serves as the template for protein synthesis and translation. So in both cases, if riboswitch in an on position, it can either allow transcription to occur or allow translation to occur. Does that make sense? That's it. Like if this structure is in the quote-unquote on position, then maybe it's a transcriptional regulation, maybe it's a translational regulation. Now imagine that result of this gene expression is some kind of an enzyme that produces some molecule, and turns out that this particular molecule can bind to a riboswitch and completely change its structure. Does that make sense? So when you change the structure of the riboswitch, you put it in the off position. And if previously riboswitch allowed transcription to proceed, now transcription stops. If previously riboswitch allowed translation to proceed, translation stops. That's it. That's like a light switch. Whatever process is regulated by a riboswitch, what's important to understand is that the trigger for going from on to off is some kind of a small molecule. And it's important not only from the standpoint of intracellular regulation of gene expressions, it can be a potential therapeutic target. If you can inhibit a particular riboswitch, you can stop transcription and translation from happening and alter, cellular function. Make sense so far? Okay. So another mechanism that, to regulate gene expression, some of you may have heard about it. This is called operons. So there are two types of operons that I want you to know. One of them is inducible, another one is repressible. So let's start with inducible operon, and I'm going to use the same example. We're going to go over what's going on using the example of lactose operon. So before we move on, I want to ask you a really stupid question. Where do you keep your forks? Say again? Okay, where's the drawer? In the kitchen. What about spoons? Same place. Knives? I have kitchen knives. Same place. Any garden tools in the kitchen? In the bedroom? No, right? The idea is that you try to keep relevant items more or less together. Does that make sense? bacteria tend to do the same. They tend to cluster genes that are functionally related. Okay? So this is an example of an operon. So in order to metabolize milk sugar, lactose, all genes that encode enzymes necessary for this process are clustered together. And these are called structural genes. So here you have DNA molecule, okay? And you have structural genes. I have no idea how many. Let's say it's 3. So those are structural genes. In front of the structural genes, you have a couple of regulatory sequences. One of these sequences here is the operator sequence. And sequence right here, we've already encountered. It's a promoter sequence. Okay, so you have promoter, operator, and structural genes. So, really simple question. When there is no lactose available, is there any need for a cell to produce enzymes that break it down? No. So, when lactose is unavailable, a molecule called repressor sits on the operator sequence. And this completely blocks the path for transcription of those genes for RNA polymerase. So you can see here, look, it's physically blocked. Does that make sense? Like it's physically blocked. RNA polymerase, the green one, it cannot move far. That's it. No transcription. Now you add lactose. And what happens is lactose binds to the repressor. As the result of this binding, look what happens. The repressor gets off the operator sequence with the lactose sitting on the side, and RNA polymerase can now proceed with the transcription. Okay? Makes sense, right? So these genes, the structural genes, are expressed. The enzymes that they encode are produced. And what do these enzymes do? They break down lactose, right? So you have lactose that is broken down to some kind of products, whatever they are, by the enzymes encoded in structural genes. So the inducible operon is called, first of all, it's called inducible because lactose induces the expression of the genes, okay? And inducible operons are regulated by the substrates of the relevant enzymatic reactions. We good so far? Okay. Second type of operons here are repressible operons. You can practically guess at this point that it is probably regulated by product, and it will be correct guess. Bless you. So in repressible operon, when sort of we start with the situation when promoter, operator, and you have RNA polymerase that binds to the promoter and proceeds with the transcription of the structural genes. Now, is there a repressor? The answer is yes, absolutely. There is a repressor. It hangs out here. Okay. Now, the structural genes, what they do, they take some reagents, whatever. Okay. And the products of the gene expression, the proteins encoded by the structural genes, they facilitate the formation of a product. In this case, it will be amino acid tryptophan. Okay, the tryptophan binds to the repressor, and what you see now is following. Now you can see your operator sequence being occupied by the repressor because tryptophan is sitting on the side of it. So RNA polymerase can't move forward anymore. And the product, look, the product of enzymatic reaction represses the expression of the relevant structural genes. Does that make sense? And it's, again, it's a classic negative feedback because look what happens. When you start supplying lactose here, you start supplying lactose, genes are being expressed, enzymes are being produced, and those enzymes break down lactose. When they break down all the lactose, nothing can remove repressor from the operator sequence. When lactose is gone, we go to the square one. With the tryptophan, the more tryptophan you have, the more repressor will be sitting on the operator, right? So it blocks the synthesis. Concentration, it's not getting synthesized, it's being used. Concentration of tryptophan drops, repressor jumps off the operator because there's no tryptophan present. The whole process starts again. Does that make sense? The Operon story. Good? Okay. Questions?